Good evening, ladies and gentlemen. Thank you very much for giving me voice. Today I will be talking about uh, our recent achievements in cellulose research. The article is published in recent number of cellulose under leadership of Laura Horse from Grenoble University and is entitled Where Influence on the Structure and the Molecular Mobility of Cellulose Diamine Complexes Studied by a Multi-Scale Experimental Approach. In this paper, the influence of diamines on the intrinsic structure and molecular mobility of cellulosic complexes is discussed. Cellulose was pre-swollen by ethylene diamine, then put in contact with a rare series of aliphatic diamines. Moreover, according to their periodicity, order or disorder phase structures were identified in successes by Clodus polarization magic angle spinning. The two-dimensional white line separation and time relaxation showed the complexes do exhibit molecules motions. The cellulose a polysaccharide is the most abundant natural molecule. The interest and the research conducted on this polymer has spanned for over almost a century. As a naturally sources biodegradable material exhibiting outstanding mechanical properties, it has found creative uses such as in structural, biomedical, technical, and packaging applications. On the other hand, because of the strong inter- and intra-chain hydrogen bonds present in its structure, cellulose cannot be dissolved by standard solvents, nor be processed by thermal methods as in glass transition due to high crystallization. This remains a major issue since cells cannot be processed as a standard thermoplastic polymer. As such physical and chemical modifications have been carried, cellulose to render this macromolecule processable, yielding both cellulose derivative materials. As of now, native or pure cellulose maize are open subject of research. A proposed means to possibly overcome this is by physically swelling cellulose by integrating molecules within its structure. In order to do so, the target molecules should be able to strongly interact with cellulose and be able to disturb hydrogen bond network of this material. Moreover, studies complexes of amines, secondary tertiaries, which showed that in mistake deal with feasibility of obtaining such a complexes of to influence on the crystalline structure, specifically crystallographic planes, as well evolution of such structure with temperature. To do so, the Professor Arnes chose a series of diamine aliphatic molecules having an increasing size ranging from 2 to 12 carbons so as to have a well-defined periodicity. Moreover, we consider such molecules as they can form two hydrogen bonds per molecule at their estimated such bridges between cellulose chains in the complex. Moreover, having the same nature as ADA, We would expect these molecules to strongly bind to cellulose and induce a similar effect on structure. Experimenter. We consider flax cellulose fibers as the source material for our measurements. Flax cellulose is partially crystalline and provides a regular structure that is capable of being characterized by X-ray diffraction. To obtain the study's cellulosic diamine complex material, this cellulose was physically complex with a series of diamine molecules. These diamides are called here MDME, except for ethylene, where N stands for number of carbon between ammonia groups and in the molecule for methylene. The diamides showed the chemical structure allowed to detect chemical structure of cellular monomer with highlighting carbons of interest for structural characterization. These properties of amines, such as melting temperature, distance between ammonia groups with each are good for modeling extending by molecule. Molecules were traced using chem sketch starter and then modeling and equilibrating of the molecules undertaken before determining the distance between ammonia amide groups for each molecule. To obtain the study's complex, cellulose was firstly dried under vacuum for 1 hour at 60 degrees. Then it was swollen by putting in the contact with ethylene diamine for 1 hour at room temperature. Afterwards, the cellulose ethylene diamine complex was put in contact with introduced at molecular status. The wall was put in a round bottom flask and heated of 20 degrees Celsius. Higher than melting temperature of given diamine, the complexation was done thus at the liquid state for the targeted diamine without any solvent. The complexation time was fixed at 24 hours, but by which time the given diamine has replaced the bonded ethylene diamine with cellulose. To characterize the inner structure of complexes, what angle where destruction were carried, using the sample was plate 22 from X-ray source, which was Philips generator, with copper alpha filter of 154 angstrom, and as the energy was set to 30 kilowatts and 20 milliamperes. The fracture was recorded and photographic fields were scanned and analyzed. All of these experiments were conducted relaxation by carbon spectroscopy measurements. The spinning measurements were conducted at room temperature, at high temperature between 70 and 100 Celsius. Such experiments were done so as to observe an eventual chemical shifting on the cellulose and diamine carbons due to an increase of molecular mobility as a function of this temperature. The rise, the two-dimensional white light separation, which allowed to identify and quantify the rigid and mobile fraction in cellulose diamine, were undertaken. The delay has a consequence that the protons located in rigid parts will relax very quickly and thus will not be able to induce a cross-polarization to the adjacent carbons. However, the mobile protons will surely relax and be able to transfer their magnetization to adjacent carbons. Finally, 12 relaxation times science were carried out to complement the study on the molecular molecularity of the cellulose diamine complexes. For these experiments, a cross-pollination between hydrogen and carbon is followed by P2 pulse and then by delay time before the acquisition is made. Measurements were conducted with cellulose MDA complexes at 23, 75, and 90 degrees. Linear extraction of diamine complexes was the room temperature, making from monoclinic structure to orthodontic during research. After cellulose diamine complexes cause the change from monoclinic to orthorhombic. Thus shown the parameters in the was linear slope value unity increase the diamine increase the cell parameter of plane is directly proportional to the diamine size and the molar stoichiometry of the complexes. It is important to note that orthodrombic structure is more symmetric than monoclinic. This confirms that result observed in literature showing that diamonds are able to modify the interscalary planes, prompting the modification of its as shown schematically. To obtain cellulose complexes were then characterized by NMR. Special interest was paid to the chemical shift region of cellulose. The obtained spectra corresponding to C4 and C6 carbons were made. The NMRs showed that in present diamonds, the peak corresponding to C4 and C6 are deshielded and that shifts carbons from 90 to 76 and C6 or 65 to 70 are modified. This shows that signal in this work a pattern is observed is overlowing. For even numbered diamonds, both order and disorder peaks observed, even order intensity being larger, and when the size diameter increases, the order peak intensity decreases. On the overhead, for all the number diamonds, the disorder peak intensity is the largest, and that when the diamond size increases, and all the peak appears at the intensity of this disordered signals decreases. The response periodicity for proposed hypothesis is based on what has been observed for even and odd number polymers, such as polyamides. For instance, it is well known that even numbered polyamers have higher glass transition and melting temperature as well as larger crystalline fractions. This is due to the fact that because of the space arrangement of polyamides, even number polymers can shift the hydrogen bond for every amide in the structure, whereas this number is cut by half for odd number polyamides. Such similar effect was observed for in liquid crystals and organic molecules being hydrogen molecular bonds. Even numbered diamonds yield a purple terad on the phase could be possible because those molecules are favored to hydrogen bond with two cellulose change at the time without modifying their canonical geometrical orientation. Odd number diamonds will have to modify the structure and twist the space onto the loss favorable orientation in order to form two hydrogen bonds at once. And more disordered structure and more easily digested. This is the case, and competing this relaxation time was changed, modified, similarly making more similar angle, making this attributing the observed this consider signature of beginning of molecular search activated by presence and this is, otherwise is rigid with decreasing relaxation time. This extending slow this resistance to temperature. This to the and fast to glass transition is more favor to break the cellulose. The series of diamonds in their structure and molecular mobility was exploited. It was shown that the presence of such molecules has a notably influence on cellular structure. For instance, diffraction experiment demonstrated that crystallographic plane of the monoclinic unit cells increases almost linearly with size of diamonds and the unit with angle diminishes. So this is the all. Thank you very much for listening me.